Yes, beta. As you know, in previous class we have done chemoosmotic photosynthesis hypothesis, where we have seen how ATP molecules are produced in the presence of enzymes. Right. After that, we have seen, as I already told, in that photosynthesis, we have done that two phases are there. Light reaction. Right. In that light reaction, what we have got? We have got two molecules or energy currency. One is ATP and other one is called NADP. These H. Only the objective of the aim of light reaction, which will be light dependent. Now that is ATP, production of ATP and production of NADPH, and which are energy currency. Now the second one is a light dark reaction, which we have to start now today. Dark reaction means we are saying dark reaction, but that is not, and that is also called biosynthetic phase, or it is also called dark reaction. Right, dark reaction or dark process you can say, and this is also called biosynthetic phase. Biosynthetic phase. Bio means living thing, synthetic means something is produced. So now by using these energy currency, here we have seen in light reaction, we have seen pigments are used, water also used, right, and sunlight is also used. Then next is the CO2 is also remaining there. Now then here in biosynthetic phase, that will be, we are using fixation of carbon dioxide. What is there? Fixation of carbon dioxide. That carbon dioxide is coming from air, right? So now that biosynthetic phase is not depending on light. They are not depending on light. Because of that, it is called dark reaction. But again, number of times I told that is called dark reaction, but doesn't mean that it will occur only in night. Okay, the dark reaction, why we are calling? Because that is not light dependent. Okay, so now then we will see just biosynthetic phase, that CO2 from atmosphere, but who is going to accept it? Right? How it will be? So first of all, you know that in that CO2, when it will come from air, now in the presence of sunlight, now then that will be having a conversion of this CO2. We have got three molecules of PGA, or it is also called phosphoglyceric acid. What is called phosphoglyceric acid, and that is having three molecules of carbon. That's why it is called 3. And now such plants where the CO2 is enter, means converting into phosphoglyceric acid, 3 carbon atoms are there, that is called C3 plants. There are two types of plants are there, C3 plants and C4 plants. Now then in C4, as the name indicates, 4 means here 4 carbon atoms. 4 carbon atoms and 3 carbon atoms are there. So on the basis of here, the first of all, after that CO2 is fixed, converting into OAA, oxaloacetic acid. Right, oxaloacetic acid means here when the plants CO2 converting into oxaloacetic acid, which having 4 carbon atoms, that plants are called C4 plants. Here we will talk about here first C3 plant, where that is at the whole, that will be this fixation of carbon dioxide. It will occur done by the Melvin Calvin, biologist were there, team is there, Melvin Calvin. Mostly that will be known as Calvin cycle. It is also called C3 cycle. C3 cycle also it is called, and Calvin cycle is also called, because Calvin biologist who discovered that cycle. So now then we will see how it will be. Cycle is called, so it means that will be in a cyclic form. As you know, cyclic form where we have started, we reach again in that same position, that is just cycle. So we will see in that. I have done already because it will take time. So I have done. That will be as a C3, I told about we just not till now what we have done. We will see light reaction. In light reaction we have got ATP and NADPH. Now the in dark reaction, we are calling it dark reaction, but it's not light dependent process, that is not depending on light. Only in that process also called biosynthetic phase. Dark reaction or biosynthetic phase, where they are depending on that cycle depending on ATP and NADPH. Right, means as previous light reaction depending on sunlight, they are depending on these energy currencies, and then energy are which energy requires for this cycle, then that will be utilized from the ATP and NADPH. Okay, so now then I told about the C3 plant where the carbon dioxide is converting into the 3 carbon atoms, that is called phosphoglyceric PGA, or it is also called phosphoglyceric acid. Now then phosphoglyceric acid. When we talk about C4 plants, in C4 plants they have got 4 carbon atoms and CO2 converting into oxaloacetic acid. Okay, so now then we will start today Calvin cycle. What will be? There are total 3 steps are there in Calvin cycle. Total 3 steps are there. The first one is there. Are you getting better? Then that is here we talk about, then red color we show that carboxylation, reduction and regeneration. There are 3 cycles are there: carboxylation means addition of carbon dioxide. Carboxylation means here carbon dioxide converting into 3 phosphoglycerate or 3 PGA, or 3 means 3 carbon atom, 3 phosphoglycerate, right, 3 phosphoglycerate. Now then that will be in that but in the presence of enzyme. First of all, one enzyme is there, that is called Rubisco. What is it called? Rubisco. Means that Rubisco means, first we should know that, that is a huge abundant. This enzyme in plant world, this enzyme is in abundance as in animal protein, collagen fibers. Now that will be also in abundant in plant and protein. When in plants we talk, these are the Rubisco enzyme. Rubisco is a ribulose biphosphate, right? Ribulose biphosphate base. Here also ribulose 1,5 biphosphate, right? This one. Now then enzyme is a Rubisco. Why it is called Rubisco? Rubisco means that is a carboxylase, C4 carboxylase, right, and O4 oxygenase, right. Carboxylase and oxygen means what will be there? If this enzyme is active in with oxygen also, with carbon dioxide also, means here when they have got in place of carbon dioxide, if more concentration of oxygen, it will bind with oxygen. When that means it will depending on the relative amount of carbon dioxide and oxygen, means it will be both. So then we are giving the name is there, that name we are calling it, that is carboxylase oxygenase. So now here C and we are using O, so Rubisco, that enzyme is Rubisco. So here in the presence of this Rubisco enzyme, then from atmosphere we are getting CO2, from soil we are getting water, then that will be, that process is called carboxylation, right? In that this enzyme is working. Now then after the conversion of this carbon dioxide, we have got 3 phosphoglycerate. This is the process carboxylation, means we can say in carboxylation we are getting 3 phosphoglycerate, or you can say glyceric acid, 3 phosphoglyceric acid. So now then we have got 3 carbon atoms. Just after this 3 carbon atoms, again the red line is show that process, means first process, second process and third process, right? Now then this phosphoglycerate will be reduced, reduction will be taking place, means reduced. So then now after reducing, this phosphoglycerate will convert it into triose phosphate. At that triose phosphate will be having sucrose or starch. This is the main aim of photosynthesis. Starch or sucrose is produced. So now then we have got after this. But what will happen here? You can see 3 phosphoglycerate when reduced. So then one ATP molecule and one NADPH molecule will be utilized. Then the breakdown of ATP molecule will convert into ATP, and during the breaking of bond, the energy will release, which will utilize in this process. NADPH also converting into NADP, because then H after breaking of this, again they are providing energy. So now then that will be, we are getting, means here one molecule of ATP and one molecule of NADPH are utilized in this second process, that is called reduction. Second process, this is the first one, this is the second one, that is called reduction. And then reduction, we are getting triose sugar, or you can say sucrose, or we are getting sucrose or starch, right? After this we will see, then sugar we are getting, then the triose phosphate regeneration. Regeneration means which enzyme? This one will be again regenerate, means again we are getting, that's why with the abundance. Now then again we are getting this one, and that will be ribulose 1,5-biphosphate regeneration. During this regeneration, when again this one will produce, again one molecule of ATP will be utilized. Here one molecule of ATP utilized here, one molecule of ATP utilized here. So total we have got 2 molecules of ATP are used in this Calvin cycle, and one molecule of NADPH also using in this Calvin cycle. So this is the Calvin, this one is the Calvin cycle. Calvin is called, but Calvin popular is the Calvin cycle, right? Where the 3 steps are there. One is called carboxylation, second one is called reduction, third one is called regeneration. Regeneration of what? This one. Here carboxylation addition with carbon dioxide, which will get, we are getting phosphoglycerate or 3-phosphoglyceric acid also it is called. After reduction, then we have got sugar, and then this one will move to the regeneration. During the whole cycle we have got one molecule of ATP here, this is one molecule of ATP, this one 2 molecules, second molecules of ATP, and then one molecule of, the total we have got in Calvin cycle, we have got from this NADPH and ATP molecules we are getting from light reaction. So this biosynthetic phase or this Calvin cycle depending on the energy currency ATP and NADP. So we will see here 2 ATP molecules are used and 1 NADPH molecules are used for this cycle. Now then this enzyme is also called Rubisco. Why it is called Rubisco? Because it will depend on the amount of oxygen or carbon dioxide which is more, it will attach with that, because they have a capacity or they are active in oxygen. It can react with oxygen also, it can react with the carbon dioxide also. So both are, which are more, carboxylation what we have written, carboxylation लिख सकते हैं या carboxylation, okay, carboxylation, it's okay. So now then we have got carboxylation means addition of carbon dioxide. So here in this biosynthetic phase, carbon dioxide is going to be fixed in sugar or starch, which is the main objective of photosynthesis. In that first phase we are getting only energy currency, in second phase we are getting food. So now then all together then we are getting, in last we are getting food from photosynthesis. But I told this one, here we have got 3 carbon atoms, here we have got 3 carbon atoms. So such plants, when the carbon dioxide is going to be converted in the 3 carbon atoms, that is called C3 cycle, which I told, right? So this is also known as Calvin cycle, is also known as C3 cycle, right? That is also biosynthetic phase, that is also called Calvin cycle, that is also called C3 cycle. Why it is called Calvin cycle? Because scientist was there who discovered it. Why it is called C3 cycle? Because we are getting conversion of carbon dioxide into 3 carbon atoms. So 3 carbon atoms means that is a C3 plants. So this cycle occurs in C3 plants. Now that is also called biosynthetic phase. Biosynthetic phase because it will be something is produced in living organisms in dark reaction by utilizing energy. So now then this is about the Calvin cycle, or you can say C3 cycle, or you can say you can say biosynthetic phase, or you can say dark reaction. Dark reaction, again I told, misnomer, means whatever the name is given, that is not fit for their function, right? We are calling it a dark reaction, but that is not occurs only in dark, it occurs in daytime also, but that is daytime also, but they are not utilizing the solar energy. Solar energy will be utilized only in light reaction. Okay, so this is about the, it is simple, very simple. Otherwise beta, in your class, in your 11th class syllabus, that is like this. The form I have taken from your NCERT. Otherwise, that is also more steps are there, but this at this age or at this stage you will learn this cycle, and you have to learn. Maybe sometime it will be question will be describe Kelvin cycle. So we have to write all these headings are there, all points are there, where the energy is used, where the ATP used, where the NADPH used, from where CO2 is there, what is this one, what is Rubisco, all we should know about this, you have to describe. But when only it should be draw Kelvin cycle. So you have to write the in that way, or it's very easy to learn. So in your book is also carboxylation we have done, reduction we have done, regeneration we have done. In last you can say, in the box is given, I want to tell that box is also, you know that sugar is there, sugar formula is there, C6H12O6, right? Means sugar formula C6H12 and O6. Means in this sugar what we have got 6 molecule of carbons. In one time we are getting only one atom, one carbon atom is there. So now how many times it will repeat? It will repeat 6 times. The cycle will repeat 6 times, then it is going to be fixed in this sugar C6H12O6. Are you getting? Means here fixation of carbon dioxide 6 times. Then we have got C6, because in one time is there only C1 carbon atom we are getting. The second time 2, 3. So we want these 6 number of carbon atom. Okay, so 6 time it will repeat this one. Then we have got C6H12O6. Just see the box is there, that I want to explain this one. This is called in, this is called out, right? In means 6 CO2. I told 6 time, I told this one 6 time that will be, so 6 time CO2 will come. In one time 1. The second time, that it means it will become in second 6 time it will be, right? And then what we have got, one glucose molecule, or you can say C6H12O6, that is a 1 molecule. 6 molecule carbon atoms will cycling in that Kelvin cycle, we have got 1 molecule of carbon atom. When we talk about ATP, can you tell me ATP how many? 6 time is there. 6 time, and then we have got in 1 Kelvin cycle we have got this one 1, this one 2, right? So now then 6 time, so it will become 12. It will become 12, and then to why it is written 18? Oh, 6 turn in the cycle, and then ATP molecules are, it means how many molecules are there? ATP, 1 this one, 1 this one. We are getting 1 and 1. 6 time is there. From 1 we have got 2 molecules. 6 time we have got 12. Just we will see that how it will be written ATP. And now then 12 NADPH. Again, because why we are writing that will be only 1. We can write here 6 only. NADP here, right, 1. And then 2 ATP, 1 and 1. So 6 time it will be fixed. Then we have got 12. This one NADP, 6 time is there. We have got 6. So now then out of 12 ATP, then we have got 12 ADP. Because after breaking of this 12 ATP, we have got 12. Now 6 NADP, then we have got 6 NADP. This is a simple what we have got from this. It is given in your books or book also. So now then we just will see 6 CO2. Means 1 6O2 enter, then we have got only 1 molecule of 2 molecules of, as you see that one we have got I am not, yes, okay, one this one, 2 molecules of ATP we are getting. Now then, in Calvin cycle, 3 molecules of ATP. 3 molecules of ATP are required probably to meet this difference. Means here, 1 molecule, 3 molecules where? We have seen that only 2. Okay, the next cycle, because in that we will start C4 cycle. In that C4 cycle we will calculate it. How it is wrong? Well, in this cycle we have got only 1 molecule and 1 there, 2 molecules, 6 times it will be repeated, 6. Now then NAD, I think convert into ATP, then it will be written. I will tell you in the next class. Okay, up to this only this Calvin cycle, 3 steps are there, enzymes are also there, and you see that why it is called biosynthetic phase, why it is called Calvin cycle, why it is called C3 cycle. In next cycle, that is a C4 cycle. Okay, we will see in 6-4 cycle what is there, and one is a photorespiration also. In next class we will see that one. Okay, so now then just revise that whatever we have done.